Despite the clarification supplied by the Chakrabarty case, the early encounters between biotechnology and the law were inevitably messy. The stakes were high, and, as we shall see in the case of DNA fingerprinting in Chapter 10, lawyers, juries, and scientists too often speak different languages. By 1983, both Genentech and Genetics Institute had successfully cloned the gene for tissue plasminogen activator, TPA, which is an important weapon against the blood clots that cause strokes and heart attacks. Genetics Institute did not, however, apply for a patent, deeming the science underlying the cloning of TPA obvious. In other words, unpatentable. Genentech, however, applied for and was granted a patent, on which, by definition, Genetics Institute had infringed. The case first came to court in England. The presiding judge, Mr. Justice Whitford, sat behind a large stack of books for much of the trial, appearing to be asleep. The basic question was whether the first party to clone a gene should be granted all subsequent rights over the production and use of the protein. In finding for Genetics Institute and its backers, the drug company Wellcome, Justice Whitford concluded that Genentech could justify a narrow claim for the limited process used by them to clone TPA, but could not justify broad claims for the protein product. Genentech appealed. Well, in England, when such esoteric technical cases are appealed, they are heard by 3 specialist judges who are led through the issues by an independent expert, in this instance, Sidney Brenner. The judges turned down Genentech's appeal, agreeing with Genetics Institute that the discovery was indeed obvious, and therefore the Genentech patent was invalid. In the United States, such cases are argued in front of a jury. Genentech's lawyers ensured that no member of the jury had a college education. Thus, what might be obvious to a scientist or to legal experts trained in science was not obvious to members of that jury. The jury found against Genetics Institute, deeming the broad-based Genentech patent valid. Not perhaps American justice's finest hour, but the case did nevertheless establish a precedent. From then on, people applied for patents on their products regardless of whether or not the science was obvious. In future disputes, all that would matter was who cloned the gene first. Good patents, I would suggest, strike a balance. They recognize and reward innovative work and protect it from being ripped off. But they also make new technology available to do the most good. Unfortunately, Stanford's wise example has not been followed in every case of important new DNA methodology. The polymerase chain reaction, PCR, for instance, is an invaluable technique for amplifying small quantities of DNA. Invented in 1983 at the Cetus Corporation, PCR, about which we shall hear more in Chapter 7 in connection with the Human Genome Project, quickly became one of the workhorses of academic molecular biology. Its commercial applications, however, have been much more limited. After granting one commercial license to Kodak, Cetus sold PCR for $300 million to the Swiss giant Hoffmann-La Roche, makers of chemical, pharmaceutical, and medical diagnostic products. Hoffmann-La Roche in turn decided that, rather than granting further licenses, the way to maximize the return on their investment was to establish a monopoly on PCR-based diagnostic testing. As part of this strategy, it cornered the AIDS testing business. And only as the patent expiration date drew near did the firm grant any licenses for the technology. Those granted have generally been to other major diagnostic companies that can afford the commensurably large fees. To create a subsidiary revenue stream from the same patent, Hoffmann-La Roche has also levied hefty charges on producers of machines that carry out PCR. And so to market a simple device for school children to use, the Cold Spring Harbor Dolan DNA Learning Center must pay the company a 15% royalty. An even more pernicious effect on the productive availability of new technologies has been exerted by lawyers moving aggressively to patent not only new inventions but also the general ideas underpinning them. The patent on a genetically altered mouse created by Phil Leader is a case in point. In the course of their cancer research, Leader's group at Harvard produced a strain of mouse that was particularly prone to developing breast cancer. They did this using established techniques for inserting a genetically engineered cancer gene into a fertilized mouse egg cell. Because the factors inducing cancer in mice may be similar to those at work in humans, this onco mouse was expected to help us understand human cancer. But instead of applying for a patent limited to the specific mouse Leader's team had produced, Harvard's lawyers sought one that covered all cancer-prone transgenic animals. They didn't even draw the line at mice. This umbrella patent was granted in 1988, and so was born the cancerous little rodent dubbed the Harvard mouse. In fact, because the work in Leader's laboratory was underwritten by DuPont, the commercial rights resided not with the university but with the chemical giant. The Harvard mouse might have been more aptly called the DuPont mouse. But whatever its name, the impact of the patent on cancer research has been profound and counterproductive. Companies interested in developing new forms of cancer-prone mice have been put off by the fees demanded by DuPont, and those keen to use existing cancer mouse strains to screen experimental drugs have likewise curtailed their programs. DuPont has begun demanding that academic institutions disclose what experiments are being performed using the company's patented onco mice. This represents an unprecedented and unacceptable intrusion of big business into academic laboratories. UCSF, MIT's Whitehead Institute, and Cold Spring Harbor Laboratory, among other research institutions, have refused to cooperate. When patents involve enabling technologies that are fundamental to carrying out the necessary molecular manipulations, the patent holders can literally hold an entire area of research for ransom. And while every patent application should be treated on its particular merits, there are nevertheless some general rules that should be observed. Patents on methods clearly vital to scientific progress should follow the precedent set by the Cohen-Boyer case. The technology should be generally available, not controlled by a single licensee, and should be reasonably priced. These limitations by no means go against the ethic of free enterprise. If a new method is a genuine step forward, then it will be extensively used, and even a modest royalty will result in substantial revenue. Patents on products, however, drugs, transgenic organisms, should be limited to the specific product created, not the entire range of additional products the new one might suggest.